So my name is Justin Baker, and I'm the scientific director of the Institute for Technology and Psychiatry. And I'm very excited to be able to introduce our last keynote session of TIPS 2023.
It's, as was suggested, it's my pleasure to now introduce Dr. Nelson Freimer, the Maggie Gilbert Endowed Chair and Distinguished Professor of Psychiatry and Genetics at UCLA, where he directs both the Center for Neurobehavioral Genetics and the Depression Grand Challenge, which you've heard about, which aims to cut the global burden of depression by half by 2050. He's authored well over 200 high-impact articles and has long been a leader in large-scale genomics and genetic investigations, including the NIMH Whole Genome Sequencing Consortium for Psychiatry Disorders and Bipolar Sequencing. His current work focuses on developing and implementing diverse strategies for scalable phenotyping of mental health disorders. And Nelson's really been a leader in the genomics field of realizing that the current way, and I imagine you'll talk about this, that we get data for genetics is very low phenotype. And so that's in many ways why we think so many of our genetic disorders all have the same GWAS for the most part. And so we've got to find a way to have scalable, better phenotyping to really connect biology with behavior. So his current work includes using electronic health records to study the genetics of serious mental illness in Colombia and in the US, and the largest ever digital phenotyping study of depression, the UCLA-Apple Digital Mental Health Study. And so we're lucky to have him today to finish the second part of this presentation. Thank you again, Nelson.
Okay, thanks, thanks, Kerry. And yeah, this talk will be related to what Marnie talked about, but from a very different perspective. From my perspective, as Kerry has just talked about, as a long-standing academic researcher in psychiatry with a specific focus on genetics and on large-scale studies. Okay, now, why am I having trouble? Okay, so here are my disclosures. And so I'm going to talk about why I think we need digital phenotyping specifically for population-level mental health research. You've heard a lot about other aspects of mental health research today. I will be talking about the UCLA-Apple Digital Mental Health Study, which Marnie introduced. And then finally, I'm going to talk about what I think is a really critical step, which is establishing standards for digital mental health research. And again, this is my viewpoint from a career in human genetics and how I think that relates to this field. So why do we need digital phenotyping for population-level research? So again, just to stick with genetics, we know that we need very large population samples to comprehensively identify the genetic contribution to psychiatric disease risk. There are a lot of projections that have been made about how large this needs to be, and this particular graph may be on the extreme end where it's talking about samples in the many millions. But I think there's really no disagreement in the field that in order to make progress, we need very, very large samples, and therefore we need scalable ways to do so. But our current, and I would say opposing approaches to establishing phenotypes for this kind of work are really problematic. And they really can be characterized, I think, as either shallow, fast, and cheap, or deep, slow, and expensive. So on the first, you might think of subjective measures, and not even the subjective measures, let's say, that get done in typical clinical research, but ones that really are eminently scalable, such as the very, very brief self-reports that many of you have probably filled out on the 23andMe site that have now been used in hundreds of thousands or even millions of individuals as the basis for genetic studies of depression and bipolar disorder and possibly other disorders, but which really consists of phenotypes that are as deep as has anyone ever told you that you've had depression. And at the other end are objective measures, most of which are non-scalable. Neuroimaging, even if you believe it does have a role in psychiatry, as one of the questions led us to ask, but it's something that nobody would ever pretend is ever going to be scalable on the level that we're talking about. So we really need something other than what we have. Another point is that the field is really desperately trying to change the paradigms for assessment and classification, and I'm sure many people here are involved in these efforts. We've tried to move from categorical classifications like the DSM shown on the left, where two individuals, each of whom get a diagnosis of depression, may have almost no features in common, according to the way we do it, to getting to quantitative or dimensional approaches like RDoC or the height-top classifications. But unfortunately, the field has not succeeded in making almost any of the quantitative behavioral phenotypes in these systems scalable. And so this really is what brought me to focus on connected consumer devices, with the idea that they really could be a provider of massively scalable quantitative phenotypes, simply because there are billions of people that are using these devices daily. And not only are they actually owning and wearing their devices, everybody actually wants to use them. I mean, most people here are using them right at this very moment. And, unfortunately, it's easy to say, well, we have all of these disease sensors, we're ready to go. But there's really a big question about how do we turn the data that these sensors provide into useful phenotypes. So on the right side of this slide, I'm showing some of the sensors that you've heard about in previous talks. And on the left side are many of the phenotypes that we're interested in, particularly with respect to mental health disorders. But for almost none of these phenotypes could we say that they're really ready to use. And this is maybe an answer to the question that was directed to Marnie, which is, although we have these devices and we have these measures, we really don't yet have them in a way that I would say they're mostly ready to use. And you've heard already from Marnie about David Moore's hierarchical framework for considering device-based phenotypes. I do think it's a really useful way of thinking about it. It not only drives the digital mental health study, which we and Apple are doing together, but it really, I think, focuses us on what actually needs to be learned at each of these levels if we're actually going to use the data from the devices to actually become phenotypes that are informative about clinical state at the same time as being scalable. Because again, the device sensors are the easy part of this story. And so really, to me, the big question is, how do we realize the scientific potential from sensor-based phenotyping? And Marnie gave some introduction to this, I think. But what I would say that we really need as a field is to really carry out 3 steps. The first is really to begin carrying out well-powered longitudinal studies. And while I think our study is better powered than most of the studies that have been done so far, it doesn't necessarily make Marnie happy to have me say that I don't think it's well-powered enough for the things that I'm interested in. We really need to learn how the device-based measures relate to the existing systems for assessing symptoms and behaviors, and that I think is really what David Moore's framework is all about. But that to me is really the biggest goal of research. And I think a third point that's really essential is that we need to evaluate the acceptability of digital assessments to research participants. And so I'm going to tell you a little bit about the work done so far in the study that UCLA is doing together with Apple, the digital mental health study. The study is still going on, so a lot of the results are not available yet to discuss, but I will discuss what results we have now that we can discuss. The design of the study was to collect 12 months of data from more than 3,000 participants using phones and watches. That is, more than 3,000 who would actually complete 12 months of study. We actually recruited more than 4,000 people at the beginning. And we really had 4 main objectives. One was to identify the relationships between device data and standard assessments. As I mentioned, I think that's really the critical step for the field. A second one, since we're doing this over 12 months, is because we really want to see if we can identify changes in individual symptoms over time. Clearly depression is something that doesn't usually exist in a static form, let's say over the course of a year. Ultimately, we're interested in identifying genetic contributions to digital phenotypes. We're really just doing this at the most superficial level at this point in the digital mental health study, and this is actually something that really UCLA is mostly interested in actively pursuing, although I know that Apple will be interested in what we learn. And then finally, we really do need to determine the acceptability to very diverse research participants of this approach. And many people have talked about this today, but also to echo what Marnie emphasized, all of the participants in this project review and authorize each data type on device. And so they not only give consent, but we can say very assuredly that everything that they've done was acceptable to them, at least at the level of them deciding to do it. So this gives some picture of the participants. They were drawn from both UCLA health system patients. They could be patients anywhere in the UCLA health system. The entire set of people who have electronic health records at UCLA were invited to consider being participants. And then, because we were also very interested in students, we reserved about a third of the study slots for UCLA students. And very importantly, this was not designed to be a study of depression. It's not a case control study of depression. It was really a study to try to understand the way in which the sensor-based measures relate essentially to the full spectrum of severity of current depression symptoms. And so as you can see here, there's really a range from healthy, mild, moderate to severe to severe. The study was designed to be demographically diverse based on both gender, age. We were very interested to make sure that this was not just a study of 18 to 25-year-olds, and also to be diverse by ethnicity, which you can see in the pie chart on the right. So I'm not going to go into great detail about the protocol itself, but why I show this slide is that it gives some sense of, really, we asked people to do a lot over a year. They essentially, not just the passive collection of data, which essentially they did, largely continuously, they gave almost an entire work week of a year per person to participating in the things we asked them to do. And so, so starting from pre-intake to remote data collection, pre-exit, exit, this just gives some sense of all the things they did. One thing that was, I think, really exciting to me is that we actually started this study during the pandemic, and it really actually showed me, I was somewhat skeptical beforehand about whether we would be able to do a study of this magnitude entirely remotely. And it's not 100% remote, but almost everything was possible to do remotely. And that in itself, I think, was one of the most interesting things we learned. This just gives a picture of the non-sensor-based, essentially, assessments, the clinical and behavioral assessments. And what I want to convey about this is to look sort of both across and down. We were interested in getting a lot of repeat measures, so that, as I said, we were interested to see how the sensor-based information aligns with changes in clinical status from let's say, scales over the course of a year. So you can see in the middle of this slide some measures that were taken very frequently over the course of the year. But also, if you look vertically, just at the sort of number of different measures that we looked at, we made a decision right at the beginning. We weren't going to get a lot of sensor measures and one depression scale, because of the fact that even in the clinical way in which we look at depression, it is so multifaceted. We really wanted to try to get as broad an understanding of how the sensor measures were related to all of these different aspects of both depression, anxiety, and stress as we feasibly could, obviously, with some consideration for participant burden. And so I just wanted to show a few slides that relate to this point. We have looked at the correlation between a number of the different measures, and for the most part, they really match with what you would expect if you were going to get a lot of different measures. For example, I mean, if you look at the top left corner, you'll see that the different depression scale measures, are highly correlated with each other. But again, we were capturing constructs through multiple approaches, in part because we recognized that they weren't all getting at the same thing. And so just to take an example of stress, we wanted to have some measures of subjective experience of stress, some measures that get at stress from the way a lot of people look at it in terms of life events. We actually didn't really like the Holmes and Ray scale as the only one, and so we sort of developed this other one of stressful life events. We also were interested in seeing whether there was some correlation with physiologic measures of stress, although we didn't do this on a very large scale, and we haven't looked at this information yet. And also through multiple time scales. So again, we were looking at, because of the fact that the sensor data themselves will capture momentary, daily, biweekly, or very longitudinal information, we were very interested in constructs that we could look at through multiple time scales as well. And so, for example, we have multiple measures that are momentary, daily, biweekly, and infrequent. And as you can see, for many of the major domains that we were interested in, we had measures in each of these different time scales. So for example, sadness is measured momentarily by, for example, EMA type of approaches, and then daily as well, and then through different scales at a more infrequent basis. Again, not surprising that there are correlations for sadness across these different time scales. We wouldn't expect for everybody that sadness is going to be continuous over the course of a year, but certainly for many people there will be a high correlation. So I've mentioned that one of the really key features of this design, both for UCLA and Apple, was to really try to understand the acceptability of this approach to participants. And so I want to present a few slides that really speak to this point. So where we are in the study is about 3/5 of the participants have now completed their 12 months. There are another close to 40% that are still in progress. It will all finish in the beginning of 2024. And overall, if we look at all of the different tasks that we ask people to do, the adherence to what we ask them to do is really incredibly high. So if you look at the different bars, really for virtually everything, over 75% of the assessments were completed, and really for the vast majority, well over 85% were completed. And we just give a comparison in the blue bar that's going across of completion rates from what we'd sort of extracted from similar studies that have been published. And adherence to the task was not just there at the beginning, but it really maintained at a pretty high level throughout the study. We can see this both with the monthly tasks, the biweekly tasks, and even the daily tasks where people were really being asked sometimes to do a lot in a particular day. And so, I mean, that gives some idea in terms of what people did in terms of, let's say, voting with their feet, but we also were interested in what their experience was subjectively. And so we asked a series of questions, and so we asked at intake and halfway through and at the end how they felt about the study experience. Most of them were very positive, and although there was some attrition in positivity over the course of the study, it was to me actually remarkably little. We asked people to log mood every day, which we thought might be found burdensome by people. But what we really didn't know what to expect is whether they would actually find it helpful in terms of their own feelings about their mental health and well-being. And really, throughout the study, the majority of people did find it helpful to do so. And again, we didn't really have an expectation about whether the study participation would have an impact on people's mental health. We were obviously not providing any treatment. Some people were in treatment. But we asked them how they felt their mental health had changed since they started the study. And again, the vast majority of people felt that their mental health had actually improved over the course of the study. And so to me, this is a really interesting result, which is if you look at the remission in terms of depression scales, that there's really a pretty high rate of remission from participating in the study. And I think that it's, I think this is a really interesting observation. And I don't know what it means, and I really think it deserves a lot more attention to understand better what it means. But I remember in one of the sessions earlier today, someone was talking about what it is that you need to do to get people to actually participate in providing information. And can you get people to provide information, let's say if you're not providing them with a lot of remuneration, to keep doing that in a research study. And I wonder whether the fact that people feel better at the end of doing a study like this may in itself be an important indicator about what we can actually expect when we try to turn the use of these kinds of tools into clinical practice. I think, to me, that's an interesting discussion point. I just want to say a couple of things about the daily and momentary mood analyses. We did two different types of things. We did daily mood assessments throughout the year where every day, one time, they were asked to rate how they felt with respect to 5 different items. And both of these were the 5-point Likert scale. And then we were interested in variation across the day, and we recognized that we couldn't do that too often, otherwise people really wouldn't do it. And so we decided to do bursts 4 times a year, where for a period, I can't remember exactly for how long we did that for, we asked people to do this 4 times a day. And this just gives some idea about the sort of the way in which people did it. When they were trying to do this 4 times a day, they had a range of times in which they could do it. Most of them did it sort of at the beginning of the interval when they were being asked to do it, but it sort of varied across these 6-hour blocks. And I don't know that the results are particularly surprising. It's not that surprising to me that, for example, energy would exhibit significant diurnal variability, and that sadness might be a little bit more stable throughout the day. And really the only reason why I show it is because I think that what will be really interesting is to be able to, when we have the sensor data, to actually be able to correlate at a very detailed level how people have responded to these questions in relationship to the essentially continuous sensor data. I'm just going to sort of talk very briefly about the approach for processing sensor data, which, we're not really very far along in the analysis, in large part because the study is still continuing. But I'm just going to present the feature generation pipeline, for those who are interested, which was largely put together by Kerry, who's a PhD student in neuroscience in my lab. And really the pipeline is based on the idea of essentially separating in a unified pipeline the different data types based on how often they come in. So for example, we have minute level variables, day level features, and multi-day features. And this just gives some sense about the different categories of variables that sort of fit in these different classifications, sort of both base variables that are very simple, sensor-based variables, and others that are fusion variables that, let's say, combine sensor data with some other information like screen time while at work. Then there are other features that are these day-level proto-features, which primarily are data that relate to sleep and circadian rhythms. And then there are the multi-day level features, and I'm particularly interested in these because these trait-like features are some of the ones that may not be very easy to do genetic studies of in other settings, but here we really can actually begin to look at some very complex potential interactions. And so again, I think that for us, the differentiation between trait-like features and state-like features is really important. So the timeline for this, as I mentioned, the data collection will end in January. We're hoping that we will complete the main analyses by sometime in the summer of next year, and certainly by this time next year, I'm hoping we will have a pretty good understanding of a lot of what we've done. So I now want to come back to what I mentioned is my own greatest interest from the use of these approaches, which is how we can begin using sensor-based data to do the really large-scale studies in genetics that we need to understand the genetic contribution to risk for psychiatric disorders. And so this is essentially just an adaptation of David Moore's framework to show that it's really at these top two levels that we're interested in for genetic study. We need to, we're already doing genetic study at the disorder level, and as Kerry mentioned, that this has sort of given us a lot of results, but it's also been problematic because it's been really hard to disentangle what is really distinct to a particular disorder versus what's really sort of the shared phenotype of severe mental illness, say. But really, it's the second level down of the quantitative traits that essentially lie between the sensors and the disorders that we've had such a hard time getting to a scalable level using other approaches besides the ones that I hope we'll be able to use in the future. And sort of that leads me to what I want to end with, which is, okay, we've shown that we can scale this to thousands. And I know there are other people here that are also scaling this kind of approach in different studies to thousands. But, billions of people are using these devices, okay? I'm not grandiose enough to say we're going to launch into a study of 2 billion iPhone users, or all smartphone users. But we do want to scale to the millions to really understand mental health disorders. And so how do we do that? And that brings me to the last part of what I want to talk about, which is establishing standards for digital mental health research. And again, this is my personal viewpoint for my career in human genetics. It's really unrelated to what we're doing in the digital mental health study. But I've been in human genetics for a long time, and I think the historical perspective can be useful, and I'm going to try to give some of that. Because the development of standards was really the decisive step in human genetics. So the Human Genome Project concluded its main phase in 2000, 2001, and the publication of the first draft genome, and that was really the enabling step for all subsequent genome-wide genetic studies of diseases and traits. But 6 years later, in 2006, there were still virtually no replicated genetic association studies for any common disorders or traits throughout healthcare. And so the NIH recognized this, having spent billions of dollars to get the genome. Was it actually ever going to get used for what the taxpayers thought it was going to be used for? and they convened a work group to try to develop standards for the field. And this is what the work group accomplished. It delineated key points to consider in genotype-phenotype association reports. Before that, anything went, and we had a lot of association reports that got a lot of attention that never could be replicated. They recommended criteria for establishing the soundness of initial association reports. They recommended criteria for establishing positive replication, and they recommended approaches for data sharing. Data sharing was something that came up in a previous keynote talk. Data sharing isn't even a question in human genetics today. Basically, since 2006, virtually everything has been shared almost, not immediately, but pretty close to immediately throughout the world. And this working group's recommendations were published in Nature, and it's actually kind of remarkable to read it. I was involved in writing it, but I didn't even remember how detailed these recommendations were. And so I don't expect anyone to look at what's specifically on here, but just to basically see, like, this group working for a period of a few months came up with this entire list. Not everything in here has been adopted, but enough of it has been adopted so that the field made use of it. And the impact has really been remarkable. So as I mentioned, in 2006, there were essentially no replicated genome-wide associations for any common disorder or trait. This slide shows all of the chromosomes, and all the colors that appear are stringently associated traits to a particular region of the genome. And so each one of these points in this time series are the accumulation of more and more associations. And I think they stopped counting in 2019. It's not that the work stopped, but essentially there was no way to display it at that point. So this really had a big impact on me, and as long as I've been working in trying to develop digital phenotyping as a tool for mental health research, I've been wanting to try to find a way to get the field to develop the same kind of consensus standards as the geneticists did more than 15 years ago. And finally, in 2023, the NIMH and the Wellcome Trust sponsored a workshop for this purpose. It was actually supposed to occur in the fall of 2020. Sort of a humorous aside is that there was an email from the person at the NIMH who was funding it saying, just wait a few months, we'll be ready to meet in person by December. So anyway, it happened in 2023. But the idea was not that this meeting was going to develop the consensus, but really that it was going to initiate the process of developing consensus. And some of the people at this meeting are here. There were more than 50 attendees from academia, community organizations, industry funders, regulators. And we had work groups which produced draft reports in 4 areas that were considered key to developing standards: digital infrastructure, data flow, study design and reporting, and a user's perspective. I'm not going to go into the details of what each of the work groups thought were the challenges in each of these areas, but just to sort of show you them by comparison with what I showed you a few minutes ago for the genetics, which is, where we are in this field is that we have these kind of broad headlines of what we need to accomplish, but what we don't have yet are the details that people can coalesce around, that funders can decide to fund, that technology companies can look at and see this is what the research community thinks it needs. And this is, really what I believe really we need to do now in each of these different areas is fill in these points with the details. Again, I don't think any of these four areas are at the point where we have the details that we need. And, and I think this is a case of last but not least, the user's perspective, the issues important to patients, providers, and communities. So this is the one area that the genetics group didn't even think about in 2006. It was not even part of the conversation. I don't think there were any representatives of any community. There may have been somebody who was a representative of some patient group, but I mean, maybe one person at most. But we're obviously in a different era in 2023, and I think many people would say this is the paramount area, and I think nevertheless, the challenge areas for this particular set of points are in some ways the greatest, and figuring out how to really get this representation. Again, I mean, it's not that hard to figure out who the academic users should be, but this is really the hardest part. So, I think that the other thing that we recognized is that there are a lot of challenges, even with the standards, for the field to achieve scalability of the type that I'm talking about. And these are just some of the things that I think are important. Clearly it's going to need a lot of resources to actually enable us to really promote participation on the scale that we want and follow through. It's going to require a lot of resources to get equitable participation across communities and populations. The populations that are most in need of this development are the ones that are the least resourced, obviously. And so if we're serious about equity here, this isn't something that's just going to happen. Somebody is going to have to decide to really put the resources in to make that happen. Again, I really believe that we're not just one order of magnitude short of where we need to be. We're probably at least 3 orders of magnitude short of where we need to be to reach the potential of the field. I'm not saying that we won't learn anything or have useful tools clinically before that. I think we'll have useful tools clinically, within a very short period of time, but that's not the full potential of this field. But I think it's also really important from my viewpoint that organizational structure to support the kind of properly scaled research we need is likely to be beyond the capabilities of most academic groups. So we're really going to need to form structures that really enable us to work together in ways that I think in this field people traditionally have not. Again, people who are peripherally involved with genetics sort of tend to joke about the level of organization of the large consortia in genetics, as to how bureaucratic and so forth they are. And they probably think that they've been in existence for decades, but really they came about very quickly after the field recognized that it needed them. And they, for the most part, functioned really well, not without some problems. And again, I think that's really what we need right now. So again, just a final summary. I really, truly believe that digital phenotyping must become one of, if not the central tool in population-level mental health research. I mean, I really firmly believe this. I don't yet have the data to say that with the conviction that I hope I will by next year. We do need not just the very large studies that I talk about in the future, but adequately scaled demonstration studies. I do think that establishing the consensus standards could have a similar impact as occurred in human genetics. And again, user perspectives must be central to this effort from the very start. So I just want to acknowledge collaborators. First, Marnie and Crane, who have been really, truly wonderful collaborators during the whole time that we've talked about and conducted this study. Many people at UCLA who were involved in the study. I want to particularly call out Eliza Congdon, shown on the top row, who is the project director for the digital mental health study and has really done an amazing job of keeping it going, again, mostly during the pandemic. And then really this is sort of thanking the team of people that work under Eliza. It's a big team. So thanks very much.
Thank you, Dr. Freimer, for an excellent talk. I think we have time for one or two questions.
Yes, thank you for an excellent talk. One quick question. The focus and a number of people have commented today that there's a real crisis in youth in mental health. And this may be a question for Marnie as well. What's the prospect for studying earlier age cohorts that have access to these devices, especially if the goal is to get longitudinal trajectories? That some of the most useful information might actually be earlier on in people's developments. And how does Apple feel about that? Have you thought about getting access to those types of data? Have you had those types of
Well, I'm definitely not
discussions?
going to answer how Apple feels about that.
Yes, yeah.
So I will answer for myself. I mean, I think your point is exactly on target. You know, I think everybody recognizes both that that's the group that is actually most likely to, you know, really actually respond to this technology in comparison with others, and also is the group that's in biggest crisis. We decided, you know, in the first instance that it was pretty difficult to do that before we understood better about how these approaches would work, but we're actually in the process right now. We've just put in a proposal to the state of California to essentially extend this idea to 16 and 17-year-olds. That's, again, not getting to the point of really initial prevention, but I do feel like there needs to be sort of a gradual accumulation of learning, which I hope will be quick.
Sure, and just to say a few more comments about collection of data from youth. For SensorKid and for Investigator Support Program, it's really a matter of your institution's IRB and then demonstrating that you have the correct oversight. We will check it, but that you have the correct oversight and are managing the younger participants in the right way. But we are supporting studies that collect data from teenagers, youth's ABC program, for example, which I think some of you are familiar with.
So I was really struck by the fact that a lot of the issues that you're grappling with, we're also grappling with on the dementia, Alzheimer's, cognitive side. And so, you know, in my talk, I talked about digital is the new blood. And that's because from the same digital data resource, we can pull out measures specific to mental health as well as cognitive health. And so one of the things that I keep being struck by is how much we all stay in our lanes, where actually if we combined across diseases around particularly digital, because if we're talking about the same sensor-based data, all we're doing is talking about pointing it to different outcomes of interest, right? So just like we use blood, raw blood, to measure multiple markers of disease, we can actually do that with digital. And that way, as we're solving some of these problems, because I've had to address exactly the same problems, including about how to deal with this across millions. So that's sort of something that I was really struck by listening to your talk.
Yeah, no, I appreciate that, and I completely agree. I mean, this is, you know, sort of the second part of my talk where I wasn't talking about the study that we're doing, but my view for the field is exactly that. I mean, that's, you know, genetics essentially, you know, you have things like very large biobank studies precisely to get away from the sort of narrow disease focus or narrow whatever focus study that existed before. And I think that's exactly where we have to get to.
Yeah, and I just, because on my side, from the Apple side, there's the Apple intuition, Biogen intuition study, using the Apple Watch, using the iPhone, collecting the sensor data, but we're staying in a little cognitive lane, whereas, wow, it'd be nice to combine it a little bit and do both.
Nelson, just a very quick question. Since this is an incredible study, what are your plans for open science for this study?
So, yeah, I mean, that's a really good question, Martin. So our plans are, to publish as quickly as we can publish things that make sense to publish, and there's no obstacles to our doing that. Obviously this is not as open as the other things that I do that are NIH studied. I mean, there's no getting away from the fact. I mean, Marty and I have a scientific collaboration, but I mean, this is a study that is sponsored by Apple, and so, I mean, we just have to be mindful of the fact that, Apple has goals beyond our open science. I don't want to say what those goals are, but I just want to say it's obvious that there are a number of considerations. I think we're going to make this as open as we can. I mean, I don't know. I don't want to make, I don't know if I put that in an awkward way, but.
Discussion for with the drinks.
One last question.
Hi there.
Last question.
I'm part of a group that is training a machine learning algorithm how to correlate oculomotor behavior with diagnoses and specifically autonomic nervous system functioning. And something that we continually run up against is how to choose what standard assessments to select, both looking to the future and what our data may be able to do, but also being able to integrate it with possibly other sensors, other data sets that we can merge with. And I was just wondering if you'd talk a little bit about best practices and approaching how you select your standard assessments with those considerations in mind.
When you say assessments, are you talking about mental health assessments now or other assessments?
Yep, the mental health assessments.
Well, I don't know that there is a best practices. We decided largely because, some of the interests of my UCLA colleagues, we decided to try to capture as much as we could the high top concepts not because we thought they were necessarily right, but because they seemed to be more comprehensive than others. Whether that's the right thing to do, I don't know that that's best practices. And again, to me, that's part of the need for this kind of standards development is actually looking to see what does ultimately associate with sensor data. I mean, that's what ultimately best practice will be the measures that when we look at them, we can say, these measures are going to be predicted by sensor data and they relate to the clinical problem that we're trying to solve. And that's the way I look at it, at least.
Thank you. All right, well, we've had an incredible day so far with excellent speakers, Dr. Bartlett and Dr. Freimer, closing out our keynotes, but the day is not over. We still have a poster session and reception in the foyer, so please join me out there with a drink and discussion of some excellent posters. Thanks.